Hello, and welcome back to our video today on DNA repair mechanisms. We're going to describe how genomic DNA is altered through damage and replication errors, and then the mechanisms that have evolved to rectify and damage these errors and damages in cells. Genomic DNA is the medium of storing all the genetic material of a cell, determining its functions it can perform, and has been conserved between all living organisms from the first forms of archaea. Therefore, DNA has been selected to be a stable storage molecule, and its structure needs to be kept intact to ensure continuity between cell lines so they can all perform their programmed function. DNA structural integrity is variable, accumulating insults from external sources, i.e. radiation or other toxins, but also intracellularly from metabolic byproducts such as free radicals or replication errors, all of which require mechanisms to rectify these and ensure mutations are not passed on that can potentially lead to a plethora of diseases. DNA damage can be categorized into exogenous or endogenous, depending on where the damage persists from, whether it's in or extracellular. These damages can manifest to different magnitudes, affecting individual bases to disrupting the DNA double helix, for example from double-stranded breaks. Exogenous agents such as ultraviolet radiation, chemical mutagens, and ionizing radiation all pose significant risks to genomic stability. UV radiation causes the formation of cyclobutyl rings from the cross-linking of adjacent T bases. That causes a kink in the DNA double helix, affecting transcription and DNA replication. Oxidative DNA damages arise from the interaction of reactive oxygen species, such as free radicals, with DNA molecules during normal cellular metabolism or due to external factors like ionizing radiation and chemical exposure. These reactive oxygen species induce chemical modifications such as base alterations, intra and interstrand cross-linking, and breaks in phosphodiester strands, contributing to genomic instability and potential mutagenesis. Spontaneous deamination, a common form of DNA damage, involves the natural removal of amino groups from DNA bases. If left unrepaired before DNA replication, these alterations can lead to base mispairing, potentially causing transition or transversion mutations, influencing the genetic information transmitted through subsequent generations of cells. The other side to this is DNA replication errors, which can arise due to various factors, such as base mismatches, low fidelity copying by specialized DNA polymerases, and insertions or deletions in the DNA. Replicative DNA polymerases exhibit intrinsic high fidelity, as they must ensure proper base pairing during nucleotide incorporation. However, occasional misincorporations can occur, and these are usually corrected by the exonuclease activity of DNA polymerases that proofread the DNA. Mismatch repair is the second level of repair if DNA polymerases don't identify a replication error. There may also be errors at replication forks and the action of topoisomerases, for example type 2 topoisomerases, making complete double-stranded breaks in the helix. Additionally, DNA replication can be continued past error sites by specialized DNA polymerases known as translation synthesis polymerases that can insert residues opposite damaged sites, proceeding along the strand and repairing damage later. However, this process increases the risk of nucleotide misincorporation, as translation synthesis or TLS polymerases lack the proofreading ability that the exonuclease activity of DNA polymerases has. Overall, DNA replication errors have the potential to impact DNA structure by introducing mutations, potentially leading to uncontrolled cell growth and development of cancer. Also, cells have distinct lifetimes, as cumulative errors in DNA replication over time contribute to the aging process and the decline of cellular function. Now looking more deeply into the single-stranded DNA mechanisms. The mechanism of base excision repair is involved in the replacement of damaged bases. DNA glycosylase enzymes read the genetic code and identify damaged bases and remove them through base excision, cleaving the glycosidic linkage to the deoxyribose. An example of this is the DNA glycosylase OGG1, which identifies 8-oxoguanine, a base mutation of guanine formed from oxidative stress. It is crucial that this base is excised as it is highly reactive and causes a base transversion that is associated with various forms of cancer. The DNA glycosylase associates with the AP1 endonuclease, which is involved in cleaving the phosphodiester bonds to remove the sugar phosphate backbone and leave an A-basic site. Reforming the A-basic site can be done through a short or long patch repair, depending on its position and cellular function. Both involve recruiting a DNA polymerase to fill in the A-basic site with a complementary nucleotide, however in different circumstances. Short patch is faster, using the DNA polymerase to synthesize one base and DNA ligase to seal the nick. Long patch involves DNA polymerase synthesizing a longer DNA strand, resulting in a flap structure that is then cleaved by the endonuclease FEN1, and the nick is sealed by DNA ligase. Nucleotide excision repair is involved in bulky lesions that distort the DNA double helix, which is typically due to UV damage. UV can cause adjacent thymine nucleotides to form crosslinks on adjacent T residues that forms dimers through a cyclobutyl ring, which distorts the DNA double helix, disrupting DNA replication and transcription. Nucleotide excision repair involves identifying the cyclobutyl ring using a DNA helicase to cause local unwinding and cleave the portion of the strand with the damaged site. DNA polymerase activity through the pCNA complex synthesizes a new strand that is joined by DNA ligase. Here, there are two processes of either global genomic, NER, or transcription coupled, NER, each of which have their own cellular role, with transcription coupled involved in ensuring gene expression, while global genomic addresses DNA damages throughout the whole genome. Looking at mismatch repair, which is involved in rectifying DNA replication errors, is a more scopic level of repair behind DNA polymerase's exonuclease activity. It utilizes the fact that the epigenetic modifications take place after a new DNA strand has been synthesized, so the mut-S enzyme can identify the newly synthesized strand from the methylated template strand, where upon binding to the damaged site, mut-S undergoes a conformational change and associates with other MMR proteins such as mut-L and mut-H. Mut-L causes DNA folding and binds to a methyl group on the template strand, while mut-H introduces a nick in the DNA strand. Exonucleases then remove the nick strand in the DNA loop, and DNA polymerase is recruited to synthesize the new strand that is sealed by DNA ligase. Mismatch repair is crucial for maintaining the fidelity of DNA replication, correcting errors that occur during DNA synthesis, preventing the accumulation of mutations, and ensuring the accuracy of the genetic code. Defects in the MMR system can lead to increased mutation rates and are associated with certain hereditary and sporadic cancers such as Lynch syndrome, characterized by an increased risk of developing certain cancers, particularly colorectal cancer. Now, moving on to looking at double-stranded DNA repair mechanisms, where you have non-homologous end joining and homologous repair. Non-homologous end joining is the process of directly ligating double-stranded breaks without the need of a homologous template. The broken DNA ends are recruited by the Ku complex to protect from degradation, and DNA protein kinases are recruited to phosphorylate other proteins involved in non-homologous end joining for alignment and ligation of DNA ligase. Non-homologous end joining allows for rapid and efficient repair of DNA breaks, contributing to the maintenance of genomic stability and protection from degradation. However, it is a low-fidelity process, and the potential for small insertions makes it error-prone and can lead to mutations. The low fidelity of non-homologous end joining is important in VDJ recombination. with mutations increasing the diversity generated by combining DNA segments from adding or removing short DNA sequences, resulting in the wide variety of antibodies that can be formed. Homologous repair is the high-fidelity variant of non-homologous end joining in double-stranded break repair, using a homologous template. The double-stranded breaks are recognized and resectioned, a process that creates a 3' single-stranded DNA overhang by the action of DNA proteins such as helicases and exonucleases. A homologous sequence is found by Rec-A in bacteria or Rad51 in eukaryotes, and the single-stranded DNA invades the homologous region of the sister chromatid, forming a displacement loop. DNA polymerase synthesizes a new DNA strand using the sister chromatid as a template. After synthesizing the new strand, the DNA structure is referred to as a holiday junction and is cleaved and rejoined to form the two different DNA strands, resulting in the creation of two separate DNA molecules. HR is a critical mechanism for repairing double-strand breaks during the S and G2 phases of the cell cycle, or when sister chromatids are available, as opposed to non-homologous end joining, which is only active in G1, where the sister chromatid is not available as a homologous template. Its high fidelity ensures the accurate repair of DNA lesions, especially in minimizing the risk of mutations. It is used alongside direct repair and translation synthesis for DNA replication to continue past DNA damage, as it is involved in restarting stalled and broken DNA replication forks. HR is also involved in the process beyond repair, such as meiotic recombination, which is essential for generating genetic diversity. In conclusion, the exploration of DNA repair mechanisms underscores the dynamic interplay between maintaining genomic stability for cellular health. DNA is susceptible to damage from exogenous factors such as radiation and chemicals, as well as errors from DNA replication. This vulnerability highlights the importance of robust repair mechanisms, and their malfunction can lead to a wide spectrum of clinical presentations, exemplified by mutations in DNA repair genes like BRCA1, which is involved in the double-stranded break repair, being associated with an increased risk of breast cancer. The intricate mechanisms of DNA repair serve as a promising target for cancer therapy. The clinical success of PARP inhibitors, exemplified by olaparib, demonstrates the effectiveness of exploiting DNA repair mechanisms. Cancer cells inactivate some DNA repair mechanisms for survival and therefore become vulnerable to drugs that induce synthetic lethality, causing selective cancer cell death while sparing healthy cells. Thank you for tuning in to our video looking at DNA damage mechanisms and also their repair. Check out our other video on DNA replication and RNA splicing if you want to look at the next steps. Thank you. Please make sure to like and subscribe.